So, welcome back, and we are going to be getting into studying more about biotechnology. We've been looking quite a bit at DNA and the human genome. So, now we're going to be looking at what it is that we can do with a human genome, how we're able to sequence the bases, and what information we can get from our genes, and so much more from that. What I wanted to share with you was this very interesting New York Times article and bringing this to the current time, talking about coronavirus. So, it's bad news wrapped up in protein inside the coronavirus genome. And so, this was published April 3rd, 2020. So, the coronavirus is a genome made up of RNA, not DNA like we have. It has RNA that's encapsulated and surrounded by all these different proteins. And we've been able to sequence our own human genome, and we've actually been able to sequence the genome for SARS-CoV-2. So, SARS-CoV-2 is the virus that causes COVID-19, which is the disease. And it's just one of a group of various coronaviruses. So oftentimes people just say coronavirus, and that's what you hear most people just say. But the actual organism, we can still debate whether or not viruses are living or non-living, but the actual structure is SARS-CoV-2, which causes the disease COVID-19. But the reason why I bring you to this article is that we've been able to, in a way, unravel this virus, which is just a string of RNA. So viruses hijack living cells to replicate and spread. So zooming in a bit, here we learned about RNA, and you see, actually, this is just the sequence of coronavirus. So the first sequence of RNA letters reads AUU, AAA, GGU, and this sequence recruits machinery inside the affected cell to read the RNA letters and translate them into coronavirus proteins. So we have talked about in our class numerous times about proteins and the function of proteins. So this New York Times article not only details SARS-CoV-2 and not just the RNA genetic information, but the proteins as well. This is the beginning of the genome. What it does is it gets into our cells and it's able to use the ribosomes within our own cells so that our own cells are able to make the proteins that the virus needs in order for it to replicate, because the virus can only replicate within a host. So here is the whole genome. Our genome has 3 billion base pairs in one of our haploid cells, or 6 billion base pairs in a diploid cell. But this is the SARS-CoV-2 genome with 30,000 RNA letters. And here is the gene for that protein, and then the gene for this protein. So we've been able to sequence the genome, but we don't necessarily know the function of this protein. In fact, we have this called mystery protein, NSP2. So scientists aren't sure what NSP2 does. The other proteins it attaches to may offer some clues. 2 of them help move molecule-filled bubbles around the cell. And so this virus has proteins. Now, this is one particular one. So part of the spike can extend and attach to a protein called ACE2. And that is on particular cells in our airways. And this is what makes it particularly fatal, as it can attack and get inside cells. So the gene, so this is the spike protein. Now, the gene for the spike protein in SARS-CoV-2 has an insertion of 12 genetic letters: CCU, CGG, CGG, GCA. This mutation may help the spikes bind tightly to human cells, a crucial step in its evolution from a virus that infected bats and other species. A number of scientific teams are now designing vaccines that could help the spikes from attaching to human cells. And that's the mutation right here. So this virus had one particular mutation, and we talked about insertions and additions. So this changed the structure of this protein, and it's a spike protein. And I said something called ACE2. What is that? So this is a really great visual, and it's COVID-19 and how it affects you. We learned about alveoli, which are these grape-like sacs in our lungs, and it's across here that oxygen may diffuse into our lungs. So as long as we are in a healthy lung, here is our capillary, and we learned about red blood cells. So in the alveolus, there's a higher concentration of oxygen. And in the capillary, there's a lower concentration of oxygen. So that oxygen can diffuse from high to low concentration and then get into our circulatory system. Now, this blood supply that gets into the lungs has a higher concentration of C2, and then that can diffuse into the alveolus. We learned that when we talked about gas exchange and transport. So that is in a healthy cell. Now, what happens is, and the role of the protein, and here we're coming back to proteins, is that on the membrane of SARS-CoV-2, which you just often hear in the news and through people speaking, it's just the coronavirus, there's a membrane protein. And that mutation of 12 bases allows it to attach to a receptor on our cells called ACE2. And then it's able to get into our cells and infect us. And once the virus is inside the cells, then there's the infection. And so the spike proteins covering the coronavirus bind to those receptors, and then that's basically when it's hijacked the cells. So throughout the course, we've been talking about proteins and structure and function. And here is how the structure of one of the proteins on the outside of this coronavirus had mutated into a way that made it more lethal. Before, it was not able to gain entry into our cells. And with that mutation, it was able to gain entry into our cells. And what's unique about this current time that we have is that we've been able to rapidly gain a lot of information with all the biotechnology resources that we have. How is it that we change the living world? How can we study the human genome? So what I shared with you was the viral genome of SARS-CoV-2, the process of genetic engineering, applications of biotechnology, and this larger field of bioethics. Now, we have been changing our world for really thousands of years. And we've been selecting for desirable traits in various plants and animals over time. So the picture that you see with the dogs, we have selected for certain traits in various dog breeds based on personality, physical features, physical traits, perhaps nose shape, ear traits, ear shape, glossy fur, temperament. So based on these desired traits, we will only allow those organisms with those desired characteristics to produce the next generation. So all of our domestic animals and most of our crop plants have been put through this process of selective breeding, in a way, really artificial selection. So the domestication of dogs and many of our domesticated animals, if you look on a deeper level, they have kind of a modified limbic system inside their brain, because one of the things that makes them domesticated is that they do not have such an adrenaline rush. So they don't have such a heightened fight or flight response. So they, when they see us, are not going to attack us. And that happens in the domestication process. A lot of our animals that are domesticated have short lifespans. They're typically animals that already exist in herds. And so that is all part of the domestication process. Now, selective breeding, this is actually a form of biotechnology because it still is an application of technological process, invention, or method to living organisms. Now, what you guys see here is, okay, I have to move this for a second. All right. So what you guys see here is two different plants. You see teosinte and you see maize. So these are two different plants, and notice the differences between them. So maize, or just corn, actually comes from this plant, and thousands of years ago, the Native Americans had basically domesticated this plant. They selectively bred for this plant. You notice that the kernels are larger, they're more yellow, and it's basically more nutritious. So it's a more productive and it's a more nutritious crop. In fact, if we try to breed teosinte and maize together, they actually do produce a viable crop, indicating that they are pretty genetically close. So essentially, what selective breeding does is it takes advantage of natural variation that exists in order to pass those wanted traits on to the next generation. But it is a slow process. It takes a number of generations in order for that to happen. But corn is one of our most important agricultural products now. And so the Native Americans had done that, but it wasn't until people would travel to the New World that that would then be discovered and brought over, or discovered, right, and brought over to European European countries. So, when we talk even further about selective breeding, there's different methods of selective breeding. So, hybridization and inbreeding. So, in hybridization, we are taking two dissimilar individuals to basically combine the best of both. So, we call these hybrids. So, when we have two individuals and take the best qualities of both. So, what you see here hidden behind this video is you see potatoes. So, one of the earliest people to hybridize potatoes is this guy named Burbank. And so, Burbank would take potatoes that had high food producing capacity and potatoes that were very resistant to disease, and he crossed them together. So, he created a hybrid. And these hybrids are often hardier than either parent, disease resistant, and capable of producing a lot of food. So Burbank would also develop hundreds of other varieties of other plants as well. But hybridization is basically taking the best qualities of two different varieties and combining them together. Now, inbreeding is a little bit riskier. In inbreeding, you are taking two individuals with similar traits and doing that in order to ensure that those desired traits are continued. The problem with that is that there could be serious genetic problems that result from it, and the greater likelihoods that they will, you know, succumb to certain genetic disorders. But there are certain points where breeders will look to increase variation. So now how can that happen? Well, one way is by inducing mutations, but specifically to bacteria. So one reason why breeders would want to introduce mutations, it can induce new traits. So mutations occur spontaneously, but breeders can increase the mutation rate by using radiation and chemicals. Breeders can often produce a few mutants with desirable characteristics that are not found in the original population. Introducing mutations has allowed scientists to develop hundreds of useful bacterial strains, including bacteria that can clean up oil spills. So the oil that we use to clean up oil spills is something that we have helped to create using biotechnology. And there are hopes that we're able to today use bacteria that we can get to clean up radioactive substances and metal pollution in the environment. Now, there are these things called polyploid plants. So we know that haploid means that there is one set of chromosomes, diploid means two sets of chromosomes, but polyploid means multiple sets of chromosomes. So here you see that there's a diploid banana and you see two sets of each chromosome. Here in the triploid banana, there's three sets of each chromosome. So the difference between these two is that the triploid banana has no seeds. So mutations in some plants produce hardier plants and plants that are larger and stronger and desirable plants that consumers might want to eat. And a lot of the foods that we eat, cotton is tetraploids, so it has four sets of chromosomes. We see bananas are triploids, so having multiple sets of chromosomes. So these are ways that breeders can increase variation. So that's enough about dogs, about bacteria, about plants. Now let's talk about us. Let's talk about the human and the human genome. I showed you guys the genome for SARS-CoV-2, and you saw that was an RNA-based genome, but we have a DNA-based genome. So we have 3 billion base pairs in a haploid cell, but 6 billion base pairs in a diploid cell. So scientists use their knowledge of the structure of DNA and its chemical properties to study and change DNA molecules. We have so much DNA that, I guess depending on font size, it's as if you could take all the DNA from you, and if you typed out all the A's, G's, C's, T's, you get like thousands of books with thousands of pages. And that's how much information is in your DNA. So scientists can use different techniques to extract DNA from cells, cut DNA into pieces, identify the sequence of bases in DNA, make copies of DNA. And in genetic engineering, biologists would be making changes in the DNA code of a living organism. Now, the DNA is this molecular substance. And if we want to cut the DNA into smaller pieces, how do we do that? How do we manipulate DNA to do what we want it to do? So there are different tools that we can use to cut, to separate, and then replicate DNA base by base in order to read the base sequences in DNA in each cell. So it seems impossible at first because we have so many, we have billions and billions of base pairs, And it does seem like a lot, but we are able to do it. Now, in order to cut the DNA, most DNA molecules are too large to be analyzed. So DNA molecules must be cut into smaller pieces for analysis. So in the 1960s, biologists discovered that there were these natural enzymes that existed in bacteria that cut up DNA. So we have an immune system, so that if anything invades our cells, we have cells within our immune system to help defend us. Except a bacterial cell does not, it's one cell. So the bacterial cell has these special enzymes that chop up foreign DNA. A bacteriophage is a virus that may invade the bacterial cell, and the bacteria has these restriction enzymes that cut the DNA as if they are just molecular scissors and to disarm this viral DNA. So here is a set of double-stranded DNA, and these restriction enzymes, the analogy is molecular scissors. You can think of them as scissors. There's all different enzymes that are able to cut the DNA. So one restriction enzyme is called EcoR1. It's called eco because it comes from E. coli. So this is a restriction enzyme that was originally in E. coli. So this restriction enzyme that's originally in E. coli recognizes the sequence GAATTC. And here it does as well. And what it does is it cuts between the G and the A. And you see it cuts between the G and the A here as well. And it cuts between the G and the A here, and between the G and the A here as well. So what we're left with is these single-stranded, overhanging pieces. So we have a single-stranded T, TAA, and here, and here as well. So those are free to stick onto other sets of DNA. Since these ends are ready to stick or bind to other pieces of DNA, we call these sticky ends. So, all in all, there are these enzymes that occur naturally in bacteria that cut DNA. They're called restriction enzymes. And they recognize these different sequences. Depending on the different restriction enzyme, it recognizes different letters. But here, EcoR1 recognizes this sequence, GAATTC, on this side and cuts between the G and the A. Here also recognized the same sequence and cut between the G and the A. And these ends are free to stick and bind. So that is how we're able to cut the DNA. So we have this molecule that we cut, and we had larger DNA fragments. So how are we able to now separate it out? Now DNA has a charge. DNA is negatively charged. So we can take advantage of the fact that DNA is negatively charged and separate it based on this charge. We use this process called gel electrophoresis. So what you see here are these different tubes, and each one has a sample of DNA. We may have already cut it up with restriction enzymes in all different sized fragments, but now we need to separate it out. These fragments may be of different sizes, and DNA is negatively charged, so we load it up at this end. Now, in this middle picture, you see that the end that it's laid at is negative, and then this end is positive. So when we turn this electric current on, then it starts to be pulled towards the positive side. And then what we end up with is these fragments that have migrated through the gel. The shorter the fragments are, the faster and further they can move. So here is the original, right? Here's the original with all the DNA. And then here in the end is what that DNA looks like after the fragments have been separated. At this end would be the longer fragments. At this end would be the shorter fragments because they traveled further. So an analogy might be like if you are using a leaf blower to move leaves over grass, the larger leaves may not move as far and as fast as the smaller leaves. So there's an analogy. And then to further that analogy, the wind that's going through the leaf blower would be like the electrical currents that's going through the gel. And then what we're left with is this banding pattern. And then we can use that to compare the genomes of different organisms and individuals, as well as identifying a particular gene in an individual's genome. This takes actually several hours to do. So there's where we load the DNA, apply the charge, and after several hours, you see the pattern. We actually apply a stain to it to see it. You can put it under UV light to see it better as well. Now, how do we read the DNA? How do we actually put all those A's, G's, C's, T's in order? So here are single-stranded DNA fragments. Okay, so there's one with a yellow T, here with a red G, here with a green A, and here with a blue C. So these DNA fragments were synthesized. Okay. Now we have an enzyme, okay, DNA polymerase, that uses this unknown strand that we're trying to sequence as a template. So it uses the template to make one new strand after another, after another, after another. So this represents the strands that we're trying to find the sequence of. And these tagged molecules are allowing us to make new fragments. So making many upon many fragments, we get many DNA strands of varying lengths, and then when we separate it out on the gel, we're able to get the sequence. Now this GIF, okay, so this GIF may be better. I like this. So here's the DNA of all different fragments. The G, A, G, C. You see all of these are different lengths. We have longer on this side, shorter on this side. So when we're trying to get this sequence, so you see 5' to 3', when we're trying to get this sequence of the DNA, those bases help us to get strands or fragments of all these different lengths. So I like this animation here. T, TA, TAG, TAGC, and so on. So all of those fragments, then when we separate it out on this gel, we're able to get the order of the DNA strand. So then that means at this end, going to this end, the order is T, A, G, C, T, G, A, T, A, C, G, A, G. And that's how that's done. So by separating it out on the gel, you can then read it. And on this side, reading it from here to up here, we have T, C, G, A, GTCAG. And that's how the sequence can be read. Now, until the 80s, researchers were doing it in that painstaking way, and it was taking a long time. But until the late 80s, we were able to use something called shotgun sequencing, using a computer program to do this a lot faster than what I was showing you there. Now, the DNA sequencing techniques to read these short fragments, all these shorter fragments can be put together to form a complete sequence through shotgun sequencing using a computer program. So what you see up here is a whole source of DNA, and it's fragmented. The computer sorts out those fragments, orders it, and overlaps particular segments in order to get the complete DNA. And down here, what I like is that you can see the large DNA. Here are all the fragments. And what the computer program actually does is it overlaps certain letters to overall put together and get the genome, to get the sequence of letters. So, cuts up the chromosomes into fragments. The fragments are sequenced, put into a computer. The computer aligns them, puts together overlapping fragments, and reads them in a complete sequence. And now, today, everything just gets faster, more inexpensive, and easier. Now, there are some techniques that just involve drawing a strand of DNA through a pore, and everything, again, is just less expensive, faster, and easier as technology develops even more so. So when we look at the human genome in 2003, the Human Genome Project, the first complete human genome was sequenced. So labs around the world now study which regions of DNA are transcribed into RNA, which ones bind to proteins, which parts of the DNA are marked with epigenetic tags. So we learned about the epigenome when we're talking about human genetics, and which parts of the DNA vary from one individual to the next. So, for example, out of all of our billions of base pairs of DNA, not every base pair in our DNA actually codes for something functional. Not every base pair gets translated into protein. What do these other stretches of DNA do? Well, and how many genes are there? So here, how many genes are there? So human cells have approximately 20,000 genes, but we don't know the function of more than 25% of those genes. Protein trafficking, cell structure and motility, transport, immunity and defense, cell growth and development, other metabolism, protein metabolism and modification, nucleic acid metabolism, cell communication, miscellaneous function, and still unknown. So when I was showing you the New York Times article, and it was saying that scientists still are trying to figure out the structure of this protein, there's still a certain portion of even our genome that we're still trying to learn more about. And again, it was only in 2003 that we actually sequenced the human genome. So, but by knowing what genes do, that does help us to learn more about how human cells and how our body functions. And we can apply that knowledge to understand more about genetic disorders, the same genetic disorders we did in the previous chapter. So how big is our genome? 3 billion sounds like a lot, but compared to other organisms, is it a lot? So take note that this scale, you see 10 to the 5th, 10 to the 6th, 10 to the 7th. Okay, so take note of that scale. The human genome has 3 billion bases, but only 2% of that actually codes for proteins. So the onion has 5 times as much DNA as ours, and lungfish have 40 times as much. Notice the scale down here. But very little of our DNA actually codes for protein. Part of the rest of our DNA is involved in the regulation of how our genes are expressed. And as we study more of our personal genome, it raises some interesting questions. Now, comparing two humans, only about one base in 1200 will not match. So these single base differences are called single nucleotide polymorphisms, or SNPs.